afternoon, class. Today we're going to cover the last section of Unit 9, which is Chapter 22, Part C. Again, we're in the topic of the respiratory system. Section 22.7, Transport of Respiratory Gases by Blood. So oxygen and carbon dioxide do not dissolve, have very limited solubilities in blood plasma. And this here is actually a problem because peripheral tissues need more oxygen and generate more carbon dioxide than the plasma can absorb and transport. But this problem is solved by the red blood cells, which remove the dissolved oxygen and CO2 molecules from the plasma and bind them, in the case of oxygen, or use them to manufacture soluble compounds in the case of carbon dioxide. So molecular oxygen is carried in blood in two ways. 1.5% is dissolved in plasma. So that means that each 100 ml of blood leaving the alveolar capillaries carries away roughly 20 ml of oxygen. And the rest is loosely bound to each iron of the hemoglobin that is found in red blood cells. So that would be about 98.5%. So recall that the hemoglobin molecule consists of 4 globular protein subunits, each containing a heme unit. Thus, each hemoglobin molecule can potentially bind 4 molecules of oxygen, forming oxyhemoglobin. So, association of oxygen and hemoglobin. So we said that each hemoglobin molecule is made up of 4 polypeptide chains, each with this iron-containing heme group. So each hemoglobin can transport 4 oxygen molecules. Oxyhemoglobin, abbreviated HbO2, is the combination of hemoglobin and oxygen. Reduced hemoglobin, also known as deoxyhemoglobin, is hemoglobin that has released oxygen, and we can see this in this reversible reaction. So what happens in the lungs and what happens at the level of the tissues. Loading and unloading of oxygen is facilitated by a change in shape of hemoglobin. Remember, hemoglobin is a protein. As oxygen binds, the hemoglobin molecule changes shape, increasing its affinity for oxygen. As oxygen is released, hemoglobin shape is again going to change, causing a decrease in the affinity for oxygen. When it's fully saturated, at 100%, that means all 4 heme groups carry oxygen. Partially saturated means that between 1 to 3 hemes carry oxygen. The rate of loading and unloading of oxygen is regulated to ensure adequate oxygen delivery to cells, and there are several factors that influence hemoglobin saturation. One would be the partial pressure of oxygen, and other factors include temperature, blood pH, partial pressure of CO2, and concentration of BPG. So let's look at the influence of the partial pressure of oxygen on the hemoglobin saturation. So just a reminder, we learned that hemoglobin is a protein and that the shape and functional properties of a protein change in response to changes in its environment. And hemoglobin, of course, is no exception. Any changes in shape that occur can affect oxygen binding. So the partial pressure of oxygen is one of those important environmental factors that affect hemoglobin. So the partial pressure of oxygen heavily influences binding and release of oxygen with hemoglobin. And the percent of hemoglobin saturation can be plotted against the partial pressure of oxygen, different concentrations. The resulting graph is not linear, but it's an S-shaped curve, and it's referred to as an oxygen-hemoglobin dissociation curve. So this graph is the oxygen hemoglobin saturation curve, or oxygen hemoglobin dissociation curve. So it's a graph that relates the saturation of hemoglobin to the partial pressure of oxygen. The binding and dissociation of oxygen to hemoglobin is a typical reversible reaction. At equilibrium, oxygen molecules bind to heme at the same rate that other oxygen molecules are being released. If you increase the partial pressure of oxygen, you shift the reaction to the right and more oxygen gets bound to hemoglobin. If you decrease the partial pressure of oxygen, the reaction shifts to the left and more oxygen is released by hemoglobin. So this graph is of this relationship. It's a curve rather than a straight line because the shape of the hemoglobin molecule changes slightly each time it binds an oxygen molecule, and this change affects its ability to bind another oxygen molecule. In other words, the attachment of the first oxygen molecule makes it easier to bind the second. Binding the second promotes binding of the third, and binding of the third enhances binding of the fourth, and so on. Okay, so let's look at figure 22.1, the oxygen hemoglobin dissociation curve. So on the left, the graph on the left that I talked a little bit about in the previous slide. So the x-axis tells you the relative amount or partial pressure of oxygen dissolved in the fluid surrounding the hemoglobin. The y-axis tells you how much oxygen is bound to hemoglobin. At 100%, each hemoglobin molecule has 4 bound oxygen molecules. Now, also with the lungs there, so in the lungs where the partial pressure of oxygen is high, about 100 millimeters of mercury, hemoglobin is almost fully saturated at 98% with oxygen. Whereas in the tissues of other organs where the partial pressure is low, around 44 millimeters of mercury, hemoglobin is less saturated. It's around 75% with oxygen. The figure on the right, the top right, where there's the other dissociation curve, shows that, so if you compare sea level versus a high altitude, so at sea level there's lots of oxygen at a partial pressure of oxygen in the lungs of 100 millimeters of mercury. The hemoglobin molecule is 98% saturated with the oxygen. Whereas at high altitude, we said already that there's less oxygen. At a partial pressure of oxygen in the lungs of only 80 millimeters of mercury, hemoglobin is still 95% saturated. In the bottom figure, we're comparing in the tissues. So the top was in the lungs. In the bottom figure, the bottom right, on the right, we're looking at in the tissues, and we're comparing at rest and for metabolically active tissues. So on the top figure there, in the resting tissues at a partial pressure of 40 millimeters of mercury, hemoglobin is 75% saturated. Only 23% of oxygen is carried by the hemoglobin is released. Whereas in metabolically active tissues, for example, when you're exercising, the exercising muscle, the partial pressure of oxygen is even lower, and at a partial pressure of 20 millimeters of mercury, hemoglobin is only 40% saturated. An additional 35% of oxygen has been unloaded for tissue use. In arterial blood, the partial pressure of oxygen is 100 millimeters of mercury and contains 20 mils of oxygen per 100 mils of blood, so 20 volume percent. Hemoglobin is 98% saturated. Further increases in the partial pressure of oxygen, as seen in deep breathing, produce minimal increases in oxygen binding. In venous blood, the partial pressure of oxygen is 40 millimeters of mercury and contains 15 volume percent oxygen. The hemoglobin molecule is still 75% saturated. The venous reserve is the oxygen remaining in venous blood that can still be used. factors can influence hemoglobin saturation. So increases in temperature, pH, like H plus concentration, partial pressure of CO2, and BPG can modify the structure of hemoglobin. Results in a decrease for hemoglobin's affinity for oxygen, and it occurs in the systemic capillaries, and it will enhance oxygen unloading, causing a shift in the oxygen hemoglobin dissociation curve to the right. Decreases in these factors shift the curve to the left. So the end result is that it decreases oxygen unloading from the blood. So figure 22.24 shows the effect of temperature on the oxygen hemoglobin dissociation curve. So we can see from this curve that changes in temperature affect the slope of the hemoglobin saturation curve. So as the temperature rises, hemoglobin releases more oxygen. As the temperature decreases, hemoglobin holds oxygen more tightly. Temperature effects are significant only in active tissues in which large amounts of heat are being generated. For example, active skeletal muscles generate heat, and the heat warms the blood that flows through these organs. As the blood warms, the hemoglobin molecules release more oxygen than can be used by the active muscle fibers. Figure 22.24b, now we're looking at the effect of partial pressure of carbon dioxide and or H plus concentration on the oxygen hemoglobin dissociation curve. So for normal blood with a pH of 7.4, and we're assuming a temperature of 37 degrees Celsius, we have that's the one in purple, okay? And the partial pressure of CO2 is 40 millimeters of mercury. For a decreased carbon dioxide or a pH of 7.6, then we have the dissociation curve, the blue one. And for an increased carbon dioxide of 80 millimeters of mercury or an H plus concentration of a pH of 7.2, we have the red curve. So in addition to consuming oxygen, active tissues generate acids that lower the pH of the interstitial fluid. So when the pH drops, the shape of the hemoglobin molecule changes. As a result of this change, the molecule releases their oxygen reserves more readily, so the slope of the hemoglobin saturation curve changes. In other words, the saturation declines. Thus, at a tissue partial pressure of 40 millimeters of mercury, hemoglobin molecules release 15% more oxygen at a pH of 7.2 than at a pH of 7.4. Okay, so other factors that influence hemoglobin saturation are BPG. So BPG is basically 2,3-bisphosphoglycerate, which is one of the, it's produced by the red blood cells during glycolysis. So it's one of those intermediate steps of glycolysis. And BPG levels rise when oxygen levels are low. As the cells metabolize glucose, they use oxygen, causing increases in the partial pressure of CO2 and H plus in the capillary blood. Declining pH, blood pH towards acidosis and increasing partial pressure of CO2 cause the oxygen hemoglobin binding bond to weaken, and this is referred to as the Bohr effect. And this is related to the curve that I showed you with the effect of pH on the hemoglobin saturation curve. So oxygen unloading occurs where needed most, basically. Heat production in active tissue directly and indirectly decreases hemoglobin affinity for oxygen, which allows an increased oxygen unloading to tissues that are active.
Cram. Well, welcome to another MedCram lecture. We're going to talk about the effect of different things on the oxygen hemoglobin dissociation curve. Okay, so we've got a red blood cell from end on. Kind of also looks like that with a little bit of a bulge in the middle, and that's because it's lost its nucleus. What this is, you have to remember, is simply a bag of hemoglobin. It's got no nucleus, it's got no mitochondria, so the only form of energy that it can do is glycolysis or glycolysis. And remember, that's where we get glucose, and it goes to, basically goes to pyruvate, and that gives off forms of ATP. This is not oxidative phosphorylation, this is substrate level phosphorylation, but it gives the cell the ATP that it needs. And that's important because there's an intermediate in this glycolysis that's actually going to do some things to our hemoglobin binding curve, which we're going to talk about next. But before we get to that, I wanted to explain to you what the hemoglobin molecule looks like. If you can kind of imagine, it's 4 different subunits that are connected to each other. Okay, and usually there's 2 alphas and 2 betas, but that's not important right now. So there's 4 binding spots for oxygen to bind to. So if it binds to the first spot, what happens is it causes a conformational shift with the next one that causes oxygen to bind more affinitively. And that causes a conformational shift with the next one, which causes the oxygen to bind even more affinitively. And finally, that causes a conformational shift that causes the last one to bind with even more affinity. And so what happens is you get something called cooperativity. The other term that they like to use in biochemistry is called allosteric interaction. In this case, it's not allosteric inhibition because it's actually making these globin molecules more apt to bind the oxygen molecule. The other thing that you might want to be aware of is sometimes they have different terms for these hemoglobin subunits. If they are not bound to oxygen, they're known as the tense form, or T, and if they get bound to oxygen, then they're known as R, or the relaxed form. The other thing that happens that you may want to know is that when an oxygen binds to this hemoglobin molecule, a little carbon dioxide molecule comes off, a little CO2, and you should probably know that that's known as the Haldane effect. Just some trivia there. So that when oxygen binds to hemoglobin, it releases CO2, and if you see the CO2 go up a little bit, that's known as the Haldane effect. But let's talk about the hemoglobin binding curve. So the way that this is represented, we've kind of talked about this before in the other lecture on delivery of oxygen, is there's a relationship between the partial pressure of oxygen in the blood and the saturation of the hemoglobin molecule. So this is saturation here, and this is PaO2, and we can take that all the way up to 100. So this is 100, this would be 50, this would be 25, 75. This is the PO2 what we're talking about here. And up to about 80, we're starting to see here that there's a kind of a curviness to this hemoglobin binding curve. And so the key points here that I want to show you is that there's this sort of a plateau area here where increasing levels of PO2 will not yield much more in terms of the saturation. So there's kind of a diminishing marginal utility associated with that. The other thing I want you to sort of notice is that if we were to shift this hemoglobin binding curve to the right, in other words, if it were to go from this point to this point, notice that in fact what you're seeing here is you're seeing the hemoglobin molecule as a whole being more apt to release oxygen. It's more apt to release oxygen. And why is that? Because at any given PO2, let's say 50 in this case, you'll see that in the blue hemoglobin binding curve has a lower saturation than the yellow hemoglobin binding curve. And so therefore, the blue hemoglobin binding curve is more apt to be less saturated at a given PO2 than the yellow hemoglobin binding curve. And that's important because what's actually happening is this thing is shifting back and forth as it goes through the bloodstream, depending on where it is. So that's kind of something that you should know. So here's a question. What are some things that are going to shift the hemoglobin binding curve to the right? And remember, these are things that make it less affinity. So the things that make the hemoglobin binding curve less affinitive to oxygen are all of the things that you would expect to find in the blood where oxygen needs to be given off by the hemoglobin molecule, and that would be in the muscles or placenta. And what are they? What do you find in the muscles? Are you going to see a high or a low pH? You're going to see a low pH because this is where lactic acid is being produced. This is where carbon dioxide is being given off, and we know that carbon dioxide is a Lewis acid. Number two, we would see a high temperature. Okay, your muscles are hot, right, when they're working, so that would shift it to the right. We already said that a high partial pressure of carbon dioxide is going to shift the hemoglobin binding curve to the right. Another thing that shifts it to the right is a molecule called DPG, diphosphoglycerate, otherwise known as 2,3-BPG or bisphosphoglycerate. This, as you may recall, is an intermediate of glycolysis, and this is where 3-phosphoglycerate goes to 2-phosphoglycerate, and that's an important step in glycolysis because as that happens, and as you have this buildup of 2,3-BPG, which is, by the way, seen elevated in pregnancy, which makes sense because in pregnancy you're going to want your hemoglobin molecule to be able to give up more oxygen to the fetus. You're going to do that. You're going to see this increased in pregnancy, and you're going to see your hemoglobin molecule giving up more oxygen to the fetus, and you're going to see this hemoglobin molecule shift to the right. Okay? Now, what are some things that you would see cause it to shift to the left? These are things that you would see in the lungs. So, for instance, in the lungs, you're breathing off carbon dioxide. You're going to have a low acidity, so you're going to have a high pH. Of course, in the lungs, you're breathing in air, which is cooler than body temperature. So generally speaking, you're going to have a low temperature. Number three, as we already mentioned, we're going to have a low partial pressure of carbon dioxide. And of course, four, we're not going to see maybe possibly as much DPG. And so you're going to see a shift to the left. The other thing that'll shift it to the left is fetal hemoglobin. So H, not A, but actually F, which is way out here. Okay? And that's fetal hemoglobin. Sucks up that oxygen like no other hemoglobin as it comes by the placenta. So that is the hemoglobin molecule and the dissociation curve. Thanks for joining us.
Carbon dioxide transport. Okay, so carbon dioxide we know is generated by aerobic metabolism in peripheral tissues, and it's transported in blood in three forms: as dissolved in plasma, chemically bound to hemoglobin, and as bicarbonate. So in dissolved in plasma, that's about 7 to 10%, and chemically bound to hemoglobin, just over 20%. So the CO2 is bound to the globin part of hemoglobin, not the heme, like the oxygen, and this is referred to as carbinohemoglobin. And we can see that in this reaction. And as bicarbonate ions in plasma, about 70%. So formation of bicarbonate involves CO2 combining with water to form carbonic acid, which quickly dissociates into bicarbonate and hydrogen ion, which we've already, I think we're very familiar with this formula. Carbon dioxide is converted to carbonic acid through the activity of the enzyme carbonic anhydrase in red blood cells. So the carbonic acid molecules immediately dissociate into a hydrogen ion and a bicarbonate ion, as we've talked about earlier. Hence, we can ignore the intermediate steps in this sequence and summarize the reaction as CO2 plus water dissociates into H plus and bicarbonate. So in systemic capillaries, after bicarbonate is created, it quickly diffuses from the red blood cells into the plasma. This outrush of bicarbonate from the red blood cells is balanced as Cl- moves into the red blood cells from plasma, and this is referred to as the chloride shift. So this figure 22.25a, we can see the transport and exchange of CO2 and oxygen. So at the level of the tissues, we have CO2 that is dissolved in plasma, CO2 that is transformed into bicarbonate and H plus, and CO2 that goes into the blood cells, and this is a very fast reaction, and combines with water, and then it becomes carbonic acid, and then it dissociates into bicarbonate and H plus, and then what happens is and then some of the CO2 is also bound to hemoglobin, as we can see there, and then the H plus and the hemoglobin combine, and then you have the Cl- that comes in, and that is called the chloride shift. In the lungs, at the level of the pulmonary capillaries, the processes occur in reverse. The bicarbonate moves into the red blood cells, while Cl- moves out of the red blood cells back into the plasma. The bicarbonate binds with H plus to form carbonic acid. The carbonic acid is split by carbonic anhydrase into carbon dioxide and water, and then the CO2 diffuses into the alveoli. Okay, so now here, figure 22.25b, we're looking at the what happens at the alveoli and the capillaries. You can see the red blood cell. So CO2 dissolved in plasma diffuses into the alveoli, and also the reaction bicarbonate plus H plus is converted to carbonic acid, which is split into CO2 and water. And then in the red blood cell, we have the unloading of CO2 and the chloride shift happening. So for each bicarbonate going in, the Cl- is moved outside into the plasma from the red blood cell to the plasma. And so we see that fast reaction of bicarbonate plus H plus that combines into carbonic acid, and carbonic anhydrase is going to dissociate into CO2 and water, which go into the alveoli. And then hemoglobin also has some CO2 that's bound to it, and it will dissociate into CO2 and hemoglobin, and the CO2 also diffuses, will diffuse. So the Haldane effect. So the amount of CO2 transported is affected by the partial pressure of oxygen. The lower the partial pressure of oxygen and hemoglobin oxygen saturation, the more carbon dioxide can be carried in blood. Reduced hemoglobin buffers H plus and forms carbinohemoglobin more easily. The process encourages CO2 exchange at the tissues and at the lungs. So at the tissue level, as more CO2 enters the blood, more oxygen dissociates from hemoglobin. And as hemoglobin and oxygen release oxygen, so as oxyhemoglobin releases oxygen, it more readily forms bonds with carbon dioxide to form carbamino hemoglobin. The influence of CO2 on blood pH. Carbonic acid bicarbonate buffer system helps blood resist changes in pH. If H plus concentration in blood rises, the excess H plus is removed by combining with the bicarbonate to form carbonic acid, which dissociates into CO2 and water. If, on the other hand, the H plus concentration begins to drop, then the carbonic acid dissociates, releasing the H plus, and the bicarbonate is considered the alkaline reserve of the carbonic acid bicarbonate buffer system. So we've already seen this when we talked about buffers in the first chapter. Changes in the respiratory rate and depth affect blood pH. So slow, shallow breathing causes an increase in CO2 in blood, resulting in a drop in the pH, whereas rapid, deep breathing will cause a decrease in CO2 in blood, resulting in an increase in the pH. Changes in ventilation can help adjust pH when disturbances are caused by metabolic factors. So breathing plays a major role in acid-base balance of the body. Hypoxia is an inadequate oxygen delivery to tissues, and it can result in cyanosis and is classified by cause. Anemic hypoxia is because there's too few red blood cells or abnormal or too little hemoglobin. Ischemic hypoxia is impaired or blocked blood circulation. Histotoxic hypoxia, it's when the cells are unable to use oxygen, as in metabolic poisons, for example, cyanide. Hypoxemic hypoxia is an abnormal ventilation due to pulmonary disease or low levels of oxygen in the air. And carbon monoxide poisoning, especially from fire, what happens is that the hemoglobin has a 200 times greater affinity for carbon monoxide than for oxygen, and victims have headaches and can become flushed.
Whether you're racing in a triathlon or doing something less strenuous, you need to breathe in oxygen to help you get energy, and breathe out carbon dioxide, a waste product. When you inhale, your diaphragm and rib muscles contract, increasing the volume of your lungs. When you exhale, these muscles relax, decreasing the volume of the lungs. When the lungs expand, air pressure in the lungs drops, causing air to flow into the lungs. When lung volume decreases, air pressure increases, causing air to flow out of the lungs. Air enters the nose or mouth, moves down the trachea, and goes into the two bronchi. Air moves down smaller and smaller bronchioles until it reaches a tiny sac, an alveolus. Each alveolus is surrounded by capillaries. Oxygen diffuses from the alveolus to the blood, and carbon dioxide diffuses from the blood to the alveolus. As blood flows through the capillary, it becomes rich in oxygen. In the blood, oxygen diffuses into a red blood cell and binds to hemoglobin, a protein made up of 4 subunits. One oxygen molecule can bind to each subunit. Oxygen-rich blood flows from the lungs to the heart, which pumps this blood to capillaries all over the body. Here, we see oxygen diffusing from a capillary's red blood cells into a muscle cell. Oxygen is used by the cell's mitochondria to produce ATP during cellular respiration. Carbon dioxide is released. How does carbon dioxide leave the body? Carbon dioxide diffuses from cells into capillaries. Some carbon dioxide stays in the plasma, the liquid part of the blood. Most carbon dioxide, however, enters red blood cells. Some carbon dioxide binds to hemoglobin. The rest is converted to bicarbonate, which diffuses into the plasma. This oxygen-poor blood flows back to the heart, which pumps it to the lungs. There, carbon dioxide diffuses from the plasma into the alveolus. Bicarbonate enters red blood cells and is converted back to carbon dioxide. Carbon dioxide is also released from hemoglobin. Carbon dioxide diffuses out of the red blood cells, into the plasma, and into the alveolus. When you exhale, air flows out of your lungs. And that's how you release carbon dioxide, get oxygen, and keep on running.
Section 22.8, Control of Respiration. So the peripheral cells continuously absorb oxygen and generate carbon dioxide. So under normal conditions, the cellular rates of absorption and generation are matched by the capillary rates of delivery and removal. Both rates are identical to those of oxygen absorption and carbon dioxide excretion at the lungs. If diffusion rates at the tissue level and alveolar capillaries become unbalanced, then homeostatic mechanisms will intervene to restore equilibrium. So such mechanisms involve 1, changes in blood flow and oxygen delivery that are regulated at the local level, and 2, changes in the depth and rate of respiration under the control of the brain's respiratory centers. So we're going to talk about respiratory rhythms that are regulated by higher brain centers, chemoreceptors, and other reflexes. So neural mechanisms, these involve neurons in the reticular formation of the medulla and pons of the brain. Medullary respiratory centers are clustered. There's clustered neurons in 2 areas of the medulla that are most important: the ventral respiratory group and the dorsal respiratory group. In this slide, we can see a mid-sagittal section of the brain and the position of the pons and the medulla. The ventral respiratory group, abbreviated VRG, is the rhythm-generating and integrative center. Recent evidence suggests forced expiration may involve a separate group of neurons than passive respiration, and it consists of a network of neurons in the brainstem that extends from the spinal cord to the pons-medulla junction. So here we see the VRG, where this group of neurons would be located in the medulla of the brainstem. So the brainstem consists of the midbrain, the pons, and the medulla. So this, these neurons that are located in the medulla set the eupnea, which is the normal respiratory rate and rhythm, which is between 12 to 15 breaths per minute. Its inspiratory neurons excite the inspiratory muscles via the phrenic, the diaphragm, and intercostal nerves, which are from the external intercostals, and the expiratory neurons inhibit the inspiratory neurons. Dorsal respiratory group, or DRG, if you go back to the figure that I showed you, they're just above the VRG. it's a network of neurons located near the root of the cranial nerve number 9. And this group integrates input from peripheral stretch and chemoreceptors, then sends the information to the VRG neurons. But much is not known about this group of neurons. Pontine respiratory centers. Pontine means from the pons. Neurons in the center influence and modify the activity of the VRG. They act to smooth out transition between inspiration and expiration and vice versa. They transmit impulses to the VRG that modify and fine-tune breathing rhythms during vocalization, sleep, and exercise. Lesions in this area of the brain lead to apneustic breathing, where a patient would take prolonged inspirations. Figure 22.26. The respiratory centers in the brainstem. So here the two components of the brainstem are the pons and medulla, and we can see that the pontine respiratory centers interact with the medullary respiratory centers to smooth the respiratory rhythm, whereas the VRG in the medulla contains the rhythm generators whose input drives respiration, and the dorsal respiratory group integrates peripheral sensory input and modifies the rhythms generated by the VRG. And so they're receiving and so they send innervations to the diaphragm and the external intercostal muscles. The origin of breathing rhythm is really not yet fully understood. One hypothesis is that there are pacemaker neurons in the VRG that control this intrinsic rhythmicity of breathing. But the most widely accepted hypothesis is that there is reciprocal inhibition of two sets of interconnected pacemaker neurons in the medulla that generates the rhythm. And each neuron set controls the other. Each neuron set controls the other to ensure rhythm. The depth is determined by how actively it stimulates the respiratory muscles. The greater the stimulation, the greater the number of motor units excited, increasing the depth of inspiration. The rate is determined by how long the center is active, and both are modified by changing body demands. So these respiratory centers in the brain are affected by chemical factors, influence of higher brain centers, pulmonary irritant reflexes, and inflation reflex. Chemical factors are the most important of all factors affecting depth and rate of inspiration. So these could be changing levels of the partial pressure of CO2, oxygen, and pH are the most important. So levels of these chemicals are sensed by central chemoreceptors that are located throughout the brainstem and peripheral chemoreceptors that are found in the aortic arch and carotid arteries. Just to remind you where the aortic arch is and the common carotid artery, these vessels that come out from the heart. So the partial pressure of CO2 is the most potent and most closely controlled. If blood partial pressure of CO2 levels rise, that's called hypercapnia, CO2 accumulates in the brain and joins with water to become carbonic acid. Then carbonic acid dissociates, releasing H+, causing a drop in pH, which increases the acidity. The increased H+ stimulates central chemoreceptors of the brainstem, which synapse with the respiratory regulatory centers. These respiratory centers increase the depth and rate of breathing, which act to lower blood CO2, partial pressure of CO2, and pH rises to normal levels. If, on the other hand, partial pressure of CO2 levels decrease, respiration becomes slow and shallow. Apnea is when breathing cessation that may occur when the partial pressure of CO2 levels drop abnormally low. Swimmers sometimes voluntarily hyperventilate to enable them to hold their breath longer. So this causes a drop in the partial pressure of CO2, which causes a delay in respiration as the partial pressure of CO2 levels need to build back up. And this can cause dangerous drops in the partial pressure of oxygen. Figure 22.28, changes in partial pressure of CO2 regulate ventilation by negative feedback mechanisms. So an increase in arterial partial pressure of CO2 is going to cause an increase in the partial pressure of CO2, which decreases pH in the brain, extracellular fluid compartment, and the central chemoreceptors in the brainstem respond to an increase of the H+ concentration in the brain. So this is an immediate response. Immediate 70% of the CO2 response. Also, an increase in the arterial partial pressure of CO2 is going to stimulate the peripheral chemoreceptors in the carotid and aortic bodies, and this instead mediates about 30% of the CO2 response. So both of these will send afferent pathways to the medullary respiratory centers of the brain, and these will send efferent impulses to the respiratory muscles to increase ventilation so that more of that CO2 is going to be exhaled. And then the arterial partial pressure of CO2 and pH levels are going to return to normal. Clinical homeostatic imbalance 2215. Hyperventilation is an increased depth and rate of breathing that exceeds the body's need to remove CO2. It may be caused by an anxiety attack, and it leads to decreased blood CO2 levels called hypocapnia, and it causes cerebral vasoconstriction and cerebral ischemia, resulting in dizziness and fainting. Early symptoms include tingling and involuntary muscle spasms in the hands and face, and the treatment is very simple. Breathing into a paper bag will increase CO2 levels, and that is going to restore the levels of CO2 back to normal. Influence of the partial pressure of oxygen. So peripheral chemoreceptors in the aortic and carotid bodies sense arterial oxygen levels. Declining partial pressure of oxygen normally has only slight effect on ventilation because of the huge oxygen reservoir that is bound to hemoglobin. So it really requires a substantial drop in the arterial partial pressure of oxygen below 60 millimeters of mercury to stimulate this increased ventilation. When excited, chemoreceptors cause the respiratory centers to increase ventilation. Figure 22.29, location and innervation of the peripheral chemoreceptors in the carotid and aortic bodies. So here we have, just where, like I showed you the picture, the carotid body, which is found in the common carotid artery, and the aortic bodies, which are found in the aortic arch. Yes. Influence of arterial pH. So pH can modify the respiratory rate and rhythm even if CO2 and O2 levels are normal. It's mediated by the peripheral chemoreceptors. Decreased pH may reflect CO2 retention, accumulation of lactic acid, or also excess ketone bodies, which are, let's say, during starvation or inadequate carbohydrate intake. The respiratory system controls attempt to raise the pH by increasing the respiratory rate and depth. In summary, for the chemical factors, rising CO2 levels are the most powerful respiratory stimulant. Normally, blood partial pressure of oxygen affects breathing only indirectly by influencing the peripheral chemoreceptor sensitivity to changes in the partial pressure of CO2. When the arterial partial pressure of oxygen falls below 60 millimeters of mercury, it becomes the major stimulus for respiration via peripheral chemoreceptors. And changes in arterial pH resulting from CO2 retention or metabolic factors act indirectly through peripheral chemoreceptors. So also there are other factors that influence the breathing rate and depth, like higher brain centers. The hypothalamic controls act through the limbic system. Limbic system is the basically our emotional brain, to modify the rate and depth of respiration. An example, the breath holding that occurs in anger or gasping with pain. And the change in body temperature changes the rate. Cortical controls direct signals from the cerebral motor cortex that bypass medullary controls. An example of that is, you know, how we can voluntary breath holding at least until the brainstem reinstates breathing when blood CO2 levels become critical. So this figure shows the hypothalamus, so the location of the hypothalamus, that's that structure in yellow, and the cerebral cortex of the brain. Another factor that influences breathing rate and depth are pulmonary irritant reflexes. There are receptors in the bronchioles that respond to irritants such as dust, accumulated mucus, or noxious fumes. The receptors communicate with the respiratory centers via the vagal nerve afferents, and they promote a reflexive constriction of the air passages, and the same irritant triggers a cough in the trachea or bronchi or a sneeze in the nasal cavity, just to get rid of that irritant. Another factor is the inflation reflex, also known as the Herring-Brewer reflex. Stretch receptors in the pleurae and airways are stimulated by lung inflation. They send inhibitory signals to the medullary respiratory centers to end inhalation and allow expiration. And they may act as protective response more than as a normal regulatory mechanism, so that the lungs do not overinflate. Figure 22.27, neural and chemical influences on brain stem respiratory centers. The higher brain, let's start with the higher brain centers, the cerebral cortex, voluntary control over breathing. These will have an effect on the respiratory center in the medulla and pons of the brainstem. Stretch receptors in the lungs also have this negative activity. Irritant receptors and receptors in the muscles and joints. Central chemoreceptors that respond to high CO2 levels via an increase in the or a decrease in the pH or an increase in the H+, and also peripheral chemoreceptors that are located in the aortic arch and the carotid arteries respond to a decrease in oxygen, increase in CO2, and an increase in H+ levels. Other receptors like pain and emotional stimuli act through the hypothalamus of the brain.